So I just presented the results of the SUMMIT study, which was a multinational, non-randomized phase 2 study of Neratinib in patients with HER2 or HER3 mutants. We looked at the effectiveness of Neratinib, which is an inhibitor of all the HER proteins in patients whose tumors had activating mutations in the HER2 or HER3 proteins. We found that Neratinib was most active in patients with breast cancer, HER2 mutant breast cancer, as well as HER2 mutant biliary and cervical cancers. And we found a relative absence of activity in colorectal cancer and bladder cancer. We think that there were really two things that were driving these differences. One is that certain tumor types appear to be more sensitive to inhibition with Neratinib, as well as different mutation types seen in different tumors. So we think there is both an effect of the tumor type that the patient has, but also the individual HER2 mutation that is seen in that patient. We didn't see any activity in patients with HER3 mutations, and that group is closed. We're moving very rapidly towards our combinations of Neratinib, either with other HER2 targeted therapy or chemotherapy or endocrine therapy, so essentially drug combinations in these same groups of patients. And we really think that's what it will ultimately require to make a big leap of efficacy in these patients. What we think we see is that different tumor types have different feedback pathways that overcome resistance to a single agent inhibitor. And this has been well documented, for example, in colorectal cancer, but is probably also operable in lung cancer and a number of other cancer types. So what we really need to understand is the unique biological mechanisms that are driving resistance to either certain mutation types or certain tumor types, and kind of overcome those in a strategic and scientifically driven way.